Hexose monophosphate pathway, abbreviated as HMP pathway, or hexose monophosphate shunt, is a very unique metabolic pathway. It generates several important compounds which are used in number of biochemical reactions in the human body. In today's lecture, we are going to discuss biomedical importance or significance of HMP shunt, and then we would see two important phases of this pathway. One is oxidative phase, in which irreversible reactions take place, and we create a special structure which is called NADPH. And then the second phase is non-oxidative phase, in which some reversible reactions occur, and we get different kinds of sugars ranging from 3 carbon to 7 carbon. So there are various synonyms for this pathway. So hexose monophosphate pathway or HMP pathway may also be referred as hexose monophosphate shunt. It is also known as 6-phosphogluconate pathway, and this is also called as pentose phosphate pathway. Intracellular site of HMP pathway is the cytosolic compartment of the cell. This pathway involves two most important phases. In the first phase, irreversible oxidative reactions take place, while the latter phase comprises reversible sugar phosphate interconversions. This pathway does not follow strict energetics, since no ATP is directly used up or generated in this cycle. So as such, in this metabolic pathway, there is no direct consumption of ATP, and there is no direct generation of ATP in this cycle. Glucose 6-phosphate acts as a precursor for this pathway. Glucose 6-phosphate can be originated from glycolytic pathway. So glucose 6-phosphate first enters into the oxidative phase of this metabolic tract, and during this phase of HMP pathway, what happens that carbon number 1 of glucose 6-phosphate gets released in the form of carbon dioxide, while 2 molecules of NADPH are also synthesized. Now, what is the biomedical significance of studying HMP pathway? The very first benefit or the very first outcome of HMP pathway, that is the production of this compound which is called NADPH. This NADPH is basically a biochemical reductant. This is also called as a reducing compound, and this is also referred as reducing equivalent. This NADPH in its reduced form is having hydrogens or protons, and it can give away its hydrogens and protons to the compound that need to be reduced, and then itself it becomes oxidized. So this is the very first significance of this pathway, that we can get NADPH from this. Then the second one, that this pathway can provide a 5-carbon sugar which is called ribose 5-phosphate or R5P. This is a 5-carbon sugar. Therefore, we can also call this pathway as pentose phosphate pathway, so that the word pentose phosphate is for ribose 5-phosphate. And this ribose 5-phosphate, it can act as a precursor for synthesizing nucleotides. Okay, this pathway can also provide or generate erythrose 4-phosphate, which is a 4-carbon sugar, and this can synthesize amino acids that are aromatic in nature, for example, tyrosine, phenylalanine, and tryptophan. It can also provide intermediates of glycolysis, for example, a 3-carbon compound, glyceraldehyde 3-phosphate. It can also give us a 6-carbon compound, fructose 6-phosphate. So this is how HMP pathway can be linked to glycolysis, and sometimes it is simply taken as alternative to glycolysis. This unique metabolic pathway can process sugars biochemically that we get from dietary sources or from the catabolism of structural carbohydrates. So any kind of sugar, most specifically if it is glucose 6-phosphate, so that can be well processed by this pathway. There are two phases of HMP pathway. The very first phase comprises irreversible oxidative reactions, while reversible non-oxidative reactions take place in the second phase of HMP pathway. Okay, in the irreversible oxidative phase of HMP pathway, which is the first phase of HMP shunt, this basically results into the production of one molecule of ribulose 5-phosphate, which is a 5-carbon sugar. Then in this phase, carbon dioxide is released and two molecules of NADPH are generated. Okay, irreversible oxidative phase of HMP pathway, which is the first part of this pathway. This is extremely of significance in the hepatocytes or in the lactating mammary glands or in the adipose tissue. What is the reason behind that? There, this pathway is used to synthesize fatty acids using NADPH. Irreversible oxidative phase of HMP pathway is also extremely important for testicles, ovaries, placenta, and even adrenal cortex, because there this pathway is used to manufacture steroid hormones using its NADPH. And this is also important in erythrocytes as well, or in RBCs, because in RBCs or erythrocytes, NADPH derived from HMP pathway first phase is used to keep glutathione in its reduced state. This is the complete overview of HMP pathway. You can see two phases here. The first phase is oxidative and irreversible, while the second phase is non-oxidative and reversible. In the oxidative phase, irreversible oxidative reactions take place, while in the non-oxidative reversible phase, there are simple sugar phosphate interconversions or intertransformations. Precursor of HMP pathway is glucose 6-phosphate. We have seen that this glucose 6-phosphate can be derived from glycolysis. It can also come from the degradation of the carbohydrates in the body. So this glucose 6-phosphate is converted into 6-phosphogluconate. And for the conversion of glucose 6-phosphate into 6-phosphogluconate, two reactions take place, and the intermediate which is formed in between is called 6-phosphogluconolactone. It is not shown here, but this is formed as a transient compound and it appears for a little period of time. So this glucose 6-phosphate is converted into 6-phosphogluconate by using two enzymes, since there are two reactions involved here. So the very first enzyme is glucose 6-phosphate dehydrogenase, which is abbreviated as G6PD, and the second name of enzyme is 6-phosphogluconolactone hydrolase. So with the help of these two enzymes or two reactions, glucose 6-phosphate is converted into 6-phosphogluconate. So during this reaction, water is used and NADP is converted into NADPH. So you can note here that you can synthesize or derive one NADH molecule in this reaction of HMP pathway. Now in the second reaction, this 6-phosphogluconate is converted into ribulose 5-phosphate, and in this reaction 6-phosphogluconate dehydrogenase enzyme is used. So this is that enzyme which catalyzes this step. Now in this reaction, carbon dioxide leaves and second molecule of NADPH is generated. So one is here and another is here. So in the first phase of HMP pathway, ribulose 5-phosphate is formed, carbon dioxide is released, and two molecules of NADPH are used. There are multiple uses of NADPH, which we are going to see later on, but the most important use of NADPH is its connection with reductive anabolic pathways. So those pathways that can generate new compounds by using NADPH as a reducing compound. So those pathways are dependent on NADPH. So in the first phase, we get NADPH, we get ribulose 5-phosphate, and simply carbon dioxide is released in this phase. First enzyme of first phase of HMP pathway, that is glucose 6-phosphate dehydrogenase or 6-GPT. This is the rate controlling enzyme or rate limiting enzyme of this pathway. So G6PD catalyzes the conversion of glucose 6-phosphate into 6-phosphogluconolactone. In the second phase of HMP shunt, reversible non-oxidative reactions occur. Now in this phase, different sugars ranging from 3 carbon to 7 carbon are synthesized. In the first phase of this pathway, we have seen that we get 2 molecules of NADPH, which are of multiple uses. But in this latter phase of HMP pathway, we get sugars which may have carbon number ranging from 3 to 7. So in the reversible non-oxidative reactions, sugar phosphate interconversions are done. For instance, in reversible non-oxidative reactions or reversible non-oxidative phase of HMP pathway, the 3 carbon sugar that you get, that is glyceraldehyde 3-phosphate. Now the type of 4 carbon sugar that you get in this phase, that is erythrose 4-phosphate. Then the 5 carbon sugar are of 3 types, and there are 3 5 carbon sugars that you can manufacture in the second phase of HMP pathway. First is ribulose 5-phosphate, then is ribose 5-phosphate, and the last one is xylulose 5-phosphate. Then the type of 6 carbon sugar that we produce in this phase, that is fructose 6-phosphate. And we can also get a 7 carbon sugar in this phase, and that is pseudoheptulose. So this is how we can get 7 carbon sugar, 6 carbon sugar, 5 carbon sugar, even 3 carbon sugar and 4 carbon sugar in this portion of this pathway. Now we're going to discuss second phase of HMP pathway, which is non-oxidative reversible. Okay, in oxidative irreversible phase of HMP pathway, we generated ribulose 5-phosphate, which is a 5 carbon sugar. So this ribulose 5-phosphate now can move in this way, or either it can move in this way. So first we are going to see reaction number 4, because we have done 3 reactions already. So in reaction number 4, this ribulose 5-phosphate is converted into another 5 carbon sugar, which is called ribose 5-phosphate. Now this reaction is catalyzed by ribose 5-phosphate isomerase enzyme. So you have to follow color scheme of carbons. Ribulose 5-phosphate is shown here in black carbons, and you can see here that carbons are 5 in number. And then when you get ribose 5-phosphate, again it is a 5 carbon compound, but here the carbons are depicted in red color. So ribulose 5-phosphate is the keto form of pentose sugar, while ribose 5-phosphate is the aldo form of pentose sugar. So ribulose 5-phosphate is isomerized to ribose 5-phosphate here. Now another molecule of ribulose 5-phosphate generated in the first phase of HMP pathway, it can also form another 5 carbon sugar that is called xylulose 5-phosphate. So this reaction that is catalyzed by phosphopentose epimerase. So ribulose 5-phosphate and xylulose 5-phosphate are epimers of each other, and both of these sugars are 5-carbon sugar or pentoses. So ribulose, one ribulose 5-phosphate is converted into ribose 5-phosphate, and another ribulose 5-phosphate is converted into xylulose 5-phosphate. Now in the sixth reaction, what happens? The 2 carbons from xylulose 5-phosphate are shifted to ribose 5-phosphate, and a 7-carbon structure is formed, which is called pseudoheptulose. So in pseudoheptulose, these 5 carbons have come from ribose 5-phosphate and 2 carbons have come from xylulose 5-phosphate. So there is funneling of 2 carbons from xylulose 5-phosphate to ribose 5-phosphate, and you get pseudoheptulose 7-phosphate. Now this reaction is catalyzed by the enzyme called transketolase, and this transketolase enzyme uses coenzyme thiamine pyrophosphate, which is also called vitamin B1. So in the reaction number 6, when xylulose 5-phosphate transfers its 2 carbons to ribose 5-phosphate and pseudoheptulose phosphate, then it is left with 3 carbons. So this 3-carbon structure now acts as glyceraldehyde 3-phosphate. So in this reaction, xylulose 5-phosphate and ribose 5-phosphate condense with each other. There is the shift of 2 carbons from xylulose 5-phosphate to ribose 5-phosphate, and pseudoheptulose is formed, while the 3-carbon structure left, it acts as glyceraldehyde 3-phosphate. Reaction number 7 takes place in the presence of the enzyme called transaldolase. Now what happens in this step? That 3 carbons from pseudoheptulose 7-phosphate, as you can see that these 2 carbon and this 1 carbon, so 3 carbons of pseudoheptulose are transferred to glyceraldehyde 3-phosphate. As you can see here, these 3 carbons are of glyceraldehyde 3-phosphate, and these 3 carbons it has taken up from pseudoheptulose. So it becomes a 6-carbon structure, which is called fructose 6-phosphate. Now when pseudoheptulose, it has transferred its 3 carbons, then it is left with 4 carbons, and the 4-carbon structures, it acts as erythrose 4-phosphate. So in the reaction number 7, pseudoheptulose transfers its 3 carbons to glyceraldehyde 3-phosphate, and you get fructose 6-phosphate, and pseudoheptulose itself becomes erythrose 4-phosphate. This pathway runs on the basis of the demand of these intermediates in the cell. If these intermediates are required anywhere in the body, then HMP pathway it would be occurring this way. For example, if more fructose 6-phosphate and glyceraldehyde 3-phosphate are required, then these can be generated from xylulose 5-phosphate. So another xylulose 5-phosphate from here, it can donate its 2 carbons to erythrose 4-phosphate, and it can become a 6-carbon sugar, which is called fructose 6-phosphate. So here you can see that these 4 carbons, which are given in red color, are derived from erythrose 4-phosphate, and these 2 green carbons are derived from xylulose 5-phosphate. So 4 carbons of erythrose 4-phosphate and 2 carbons of xylulose 5-phosphate makes a 6-carbon structure, which is called fructose 6-phosphate. Now when xylulose 5-phosphate has transferred its 2 carbons, then it is left with 3 carbons, and the 3-carbon structure is now acting as glyceraldehyde 3-phosphate. So with the help of xylulose 5-phosphate and its condensation with erythrose 4-phosphate can generate fructose 6-phosphate and glyceraldehyde 3-phosphate. And this reaction is also catalyzed in the presence of transketolase enzyme that uses vitamin B1 as a coenzyme. So in this phase of HMP pathway, we get many sugars. For example, we get sugars ranging from 3 carbon to 7 carbon. You can spot all these sugars in this diagram. The 3 carbon sugar is glyceraldehyde 3-phosphate. The 4 carbon sugar is erythrose 4-phosphate. 5-carbon sugar is ribose 5-phosphate. There is another 5-carbon sugar, xylulose 5-phosphate, and ribulose 5-phosphate is also a 5-carbon sugar. Then the 6-carbon sugar is fructose 6-phosphate, and pseudohaptulose 7-phosphate is a 7-carbon sugar.